So, introduction. So some of this will be review, but it should consolidate a lot of things that we talked about you watched in the videos. So, let's see. Oh, there we go. DNA fingerprinting is the same thing as DNA profiling, same word. Has nothing to do with your fingerprints. So when you see fingerprinting, it doesn't have anything to do with that. They just call it that because it's unique to you. Remember, you can slow these slides down if you need to and pause the video and copy into your document. So DNA stands for deoxyribonucleic acid. It's a mouthful, but I'm sure you can pronounce it. DNA. That's where they get the acronym from. Ain't rocket science. DNA is deoxyribonucleic acid. Only identical twins have exact DNA. And even in identical twins, they can actually have there's rare instances where they can actually have different DNA, depending on certain things that happened when they were conceived. Since the 1980s, DNA evidence has been used to investigate crimes, establish paternity, who is the baby daddy, and identify victims of large-scale disasters, such as plane crashes, such as 9/11 and the Twin Towers. So before 1980, we really couldn't use DNA evidence because the science wasn't there to extract it and use it. DNA evidence can be traced back to one and only one person. That's why they call it individual evidence. Similar to fingerprints and DNA, pretty much it. It can link a suspect to a crime. It can eliminate a suspect. And it can identify a victim even without a body. So if they have the DNA, they might be able to identify the victim even without the body. Remember, you can pause this. If we're going too fast, just pause it. Hit the pause button. The history of biological evidence in forensics. Biological evidence, as the name shows, includes things such as skin, blood, saliva, urine, semen, hair. So there's some examples of biological evidence. So basically anything that comes from a living organism. Most foreign techniques originally developed for medical diagnosis and treatment. So most of these techniques were originally developed for medical diagnoses and treatments. Karyotyping is the analysis of chromosomes. Blood types: AB, AB and O. So those are the 4 blood types of humans. Blood types are an inherited trait. Means you got them from your mom and your dad. They're used for paternity testing. And blood type testing is easier to do than DNA testing. So it's usually one of the preliminary things they do because they can rule somebody out. So remember, if somebody committed a crime and the blood found, say, of the suspect was type O, but they found type A blood at the crime scene, but they knew it was his blood or the suspect's blood, they know they could rule that person out. Structure and function of DNA. DNA is the blueprint of life. It has all the information needed to make you. It's the genetic material of the cell. It's made up of chromosomes. It has information to make proteins and replicate itself. There's what it looks like. That's a good representation. There are two backbones, and they form a spiral helix. And the rungs on the ladder would be the nitrogenous bases, such as A, T, C, and G. The backbone is made up of phosphate and sugars. So it's made up of two strands. That's why they call it a double helix. Double, two, helix because it's spiraled. Watson and Crick were the ones that described the structure of the DNA. So they based it on information they had, as well as other people, such as Rosalind Franklin, who actually provided them with the key to figuring it all out. She provided them with a X-ray crystallography of a photograph of the DNA structure that she took. So the backbone is made up of sugar and phosphate. The rungs are made up of nitrogenous bases. The nitrogenous bases being adenine, thymine, cytosine, and guanine, A, T, C, and G. DNA has four nitrogenous bases: adenine, cytosine, guanine, and thymine. Two of these are purines, two of these are pyrimidines. If you're ever asked which is which, pyrimidine has a Y in it. Cytosine has a Y in it. Thymine has a Y in it. They're the pyrimidines. Adenine and guanine are the purines. Pairing of complementary bases. A always pairs with T. C always pairs with G. Always. So if this were a segment of DNA, ACCTAGCTA, the complementary strand would be TGGATCGAT. So if you were given one of these, you should be able to provide the other. A always goes with T, C always goes with G. Humans have 23 pairs of chromosomes for a number of 46 total in the nucleus of most of your body cells. The only body cells that don't have it are your red blood cells. They have no nucleus, they have no DNA. So when they say that you can test blood for DNA, it's because you're testing the other parts of the blood, like the white blood cells and pretty much white blood cells. Each egg and sperm has half that, or 23 total chromosomes. So each egg has 23 chromosomes. Each sperm has 23 chromosomes. 23 plus 23 equals 46. So that's where we get the terms diploid and haploid. So your egg and sperm cells are haploid cells, and the somatic cell, which is a body cell, has 46 chromosomes, and that is a diploid cell. So the baby, half of the DNA comes from mom, half of it comes from dad. So literally, half of your DNA came from your mom, half of it came from your dad. There are two types of DNA. There's nuclear DNA. It's in most of your cells of your body, again, except for your red blood cells. And mitochondrial DNA. It's in the mitochondrion of the cells. So we talked about this before when we did hair. The hair shaft can contain mitochondrial DNA, and that is traced back through your maternal line, which would be your mother. Nuclear DNA is would be found in cells in your body, including the roots of your hair. But again, mitochondrial DNA would be found in just the mitochondrion. Mitochondrial DNA is circular, unlike nuclear DNA, and it's only inherited from your mother, none from father. So we still go with that. It's inherited from your mother. There are rare instances where they found that it was inherited from the father, but we will stick with it's inherited on the maternal line from your mama. Genes and alleles. Each chromosome contains many genes. These are genes on the chromosome, not like genes like Levi's and Wrangler. Genes, the DNA sequences that have instructions that determine our inherited traits or characteristics. Examples, your blood type, your hair color, your earlobe shape, things like that. Things that are inherited from your parents. An allele is one or more alternative forms of a gene. So we'll simplify it to the point where like you have two genes, one on each strand of DNA. You could have an allele for eye color. Eye color is pretty complex, but you could say one of those genes could be for blue eyes, one could be for brown eyes. But eye color is a little bit more complicated than that. But an allele is just the same gene, same location, but a different form of that gene. So it could be eye color blue versus brown, hair color brown versus black. But an allele is just a different form of the same exact gene. One allele comes from mom, one comes from dad. Examples, gene codes for normal hemoglobin while another codes for abnormal hemoglobin. So hemoglobin is the iron-containing pigment in your red blood cells. So you have a gene on one strand of DNA. The gene would be a little segment of that DNA. So you have one that codes for, well, they both code for hemoglobin, some type of hemoglobin. One might code for normal, one abnormal. So again, they're alleles of each other, but they're just different forms of the same gene. Human genome is the total amount of DNA in a cell. There are about 6 feet of DNA in each of your body cells. DNA is in chromosomes. DNA is in mitochondria. This is the total amount of DNA in a cell. There are 3 billion base pairs of DNA in each human cell. 3 billion base pairs. That means there's 3 billion pairs of ATCGs in each human cell. There's a lot of it. There are 23,688 encoded genes, each with 3,000 base pairs. So you have about 24,000 genes, each containing an average of about 3,000 base pairs. This is only about 1.5% of the entire DNA in your genome. So there's a lot of DNA that does not encode for anything. So 98.5% of your DNA is non-coding DNA and does not make any protein. They used to refer to this as junk DNA. Now they refer to it as a non-coding DNA. So we know it doesn't code for anything, but to say it's like junk, like it's worthless, is not a good term. So 98.5% of your DNA does not code for any proteins. That's a lot. A VNTR is a variable number of tandem repeats. So you might have this repetition where it goes CAT, AC, AG, AC, CAT, AC, AG, AC, AC. So you might have 4 repeats, somebody else might have 8, somebody else might have 12, somebody else might have 2. So there's a variable number of tandem repeats. So this person has 4, this person has 7. So this is a way to differentiate between different people's DNA. So the repeat would be this segment. So here there's 4 repeating segments. Here there's 7. And I think that's it for right now. So catch up on your notes.